Good morning, everyone. We're going to get started on the 5th Annual ALS Symposium. I want to thank everyone for being here today. My name is Maureen Brinzer, and I'm the Executive Director for the ALS Association Northern New England Chapter, and I am so thrilled to have all of you here. We have a packed agenda with a lot of great information to share with all of you. Today's symposium is a year in the making. Amber Stalker, who is our Director of Care Services, received this as her first major project when she joined us last December. She has worked tirelessly in building this program and that will provide you all with the latest information and research as well as education on a variety of topics that we get questions about all of the time. She has also built an impressive exhibitor area for attendees to visit and gather more information. Amber has done an absolutely phenomenal job, and I would like to take a moment to recognize her as she walks out of the room for all of her hard work and commitment. So one of the keys to the success of today's symposium, is a committee that Amber has built in creating today's symposium. The committee is comprised of volunteers located in New Hampshire and Maine and Vermont, and they have met over the course of 11 months on a monthly basis to help identify and recruit speakers, exhibitors, volunteers, as well as develop a strong educational program. I'd like to take a minute to thank each one of our committee members. Tammy Dustin is back there. Thank you, Tammy. Kate Spanier. I just saw her there's Kate right there. Thank you, Kate. Dr. Greg Cox is right here. Tom Farrell is in the back of the room. Oh, no, he's in the front of the room now. Thank you, Tom. And Carol Allen, who is not here today, also helped us, and Carol is from Vermont. I'd like to take a moment to introduce you to the Northern New England team. I have the honor and the pleasure to work with the best team ever. I am so proud to come to work every single day and work side by side with this team. They are a dedicated group of people that I know that I can rely on each and every day. We are a lean team, but we are mighty. So I'd like to take a minute to introduce you to my partners in crime. In the back of the room is Lisa Pillsbury, who is our office administrator. Also next to Lisa is Carla Wheaton, who is our event manager for the state of Maine. Shauna Zeckman is also in the back of the room. She is our event manager for New Hampshire and Vermont. You all met Amber Stalker, who is our Director of Care Services and our New Hampshire Care Coordinator. Right next to Amber is Lori McFerrin, who is our Care Coordinator for the state of Maine. And the newest member of our team is Karen Hammer, who is our Care Service Coordinator in Vermont. Thank you. I am honored. You are all the best. I'd like to give a special shout out to our sponsors. First, our presenting sponsor today is Heron & Smith, and you'll hear a little bit more about Heron & Smith. But in addition to Heron & Smith, we do have Mitsubishi Tanabe Pharma America, as well as the medical store. So let's give them a round of applause, too. Lastly, I want to thank all of the speakers and the exhibitors that are here today. We are so appreciative of your commitment to coming to this fantastic day. We couldn't do it without your partnership, so thank you all. So, as I mentioned earlier, this is the 5th Annual ALS Symposium for the Northern New England Chapter. It's changed a lot over the years. For a few years, we held it at Lebanon at Dartmouth-Hitchcock Medical Center. One year it was right here in this very room. One year we tried to split it out into each individual states. It's really tough, though, because the Northern New England chapter covers Maine, New Hampshire, and Vermont, which is such a vast territory. that it's not convenient for everyone to attend. So, for the first time, we are offering education that is via live stream. So that for those that cannot attend, they are actually here today, live stream, which is really kind of cool. So in addition to the about 100 people that are here, we have people participating from their own remote home settings. I do have a huge favor to ask all of you, though. In all of your bags, you've received evaluation forms. And I am going to ask that all of you, not now, don't fill them out yet, but before the end of the day, before you leave today, please fill out the evaluation forms, because those are so valuable to us. We will take those and we will plan next year's symposium based on that. And I believe we're going to have the evaluation available for the online. It's going to be attached so that they can just click on it and fill it out. So anybody that is listening to us live stream, you will be able to also get the evaluation form. So, and I'm thinking of scheduling a meeting with Amber tomorrow so that we can start planning 2019. What do you think, Amber? So also included in your bag is the agenda, and we will be offering 4 presentations here in the Salon AB. We will be starting our research presentation and panel discussion in just a few minutes, but also in this room will be insurance conundrums at 11:05, advocacy at 1:05, and message and voice banking at 1:25. And these are the programs that also will be live streamed. All of you in this room will have the opportunity to go into breakout sessions of your choice. Unfortunately, we can't do that for the live stream, but if it is successful, maybe that is something we can consider. What's that? Calories, I'm sorry, okay, and another one, calories count. I apologize. So there's going to be some here, but there's also going to be some breakout sessions. The breakout sessions are not live streamed, though, so unfortunately, we could not make that happen for this year, but maybe for next year. The other piece of paper that you received in your bag is a bingo game, and it's what we're encouraging everybody to do. We want you to visit the exhibitor tables and talk to them, because they have a wealth of information to share. And if you take your bingo cards and you have 12 of them, fill it out, you can turn it in, and we will be giving away a raffle at the end of the day. So make sure you do that. Okay, before I get the hook to stop talking here, I do have a few housekeeping items. Please turn off your cell phones or put them on vibrate. Restrooms for anyone are down the hall. You kind of go down the hall and take a left. They're down there. And for those of you that need a quiet place to go, we do have a serenity room that is down the hall right outside these doors. You just follow the hotel entrance hall. And don't forget to complete those evaluations. Okay, so enough of me. I would like to introduce to you our presenting sponsor, Tammy Dustin, who is the CEO and president of Heron & Smith. I have the honor and privilege to work with Tammy often. Heron & Smith is one of our durable medical equipment providers here in New Hampshire who goes above and beyond the call of duty for our chapter as well as for those that have been impacted by ALS. Tammy also serves as a member of our advisory council and is a huge asset to the team. So without further ado, I would like to introduce Tammy Dustin.
Good morning. I will keep this brief so we can keep the agenda on schedule. So Heron & Smith has been fortunate. We've been a partner with the ALS Association for over 10 years. And within the past 5 years, the scope of the association, the advocacy, the commitment, the passion with the staff has been outstanding. And Heron & Smith has been fortunate to where, with the wonderful staff, to educate, help them with advocacy, provide a loaner closet throughout the state for those impacted with ALS that need equipment. So I just want to say thank you to Morit and her excellent staff. We couldn't be more proud to work with the ALS Association. We're proud to be a corporate sponsorship. We will continue to do so. And please feel free to visit our room. It's down the hall to the right. There's several pieces of medical equipment set up, or if you want to have a one-on-one with certain situations or questions, please feel free to seek us out. And thank you again, and enjoy the day. It's going to be a wonderful, wonderful sessions. Thank you.
Okay, so we're going to get going on the research, and I need to find Dr. Cox's bio. So I'm going to introduce to you Dr. Cox. Dr. Gregory Cox received his Bachelor of Science degree in biology from Humboldt State University in California and a PhD in human genetics from the University of Michigan. He arrived at the Jackson Laboratory at the end of 1994 to do his postdoctoral training with Dr. Wayne Frankel, studying the genetics of epilepsy and neuromuscular disorders. He is currently an associate professor at Jackson Labs. Dr. Cox's research program is focused upon molecular pathways necessary for the normal function and survival of motor neurons and their skeletal muscle targets. Specific areas of study include mouse models of spinal muscular atrophy with respiratory distress, amyotrophic lateral sclerosis, muscular dystrophies, and a spectrum of motor neuron diseases and peripheral neuropathies. Without further ado, Dr. Greg Cox.
A long introduction for me just so that I get to introduce our next speaker. So as of this morning, I just met Jill Yursak. She is, I have to look up her title because I suck with titles. She is the Director of Mission Strategy, the ALS Association. But I know her research anyway because she has a research background and she has been in the neuromuscular field getting her PhD at Thomas Jefferson University, where she worked on spinal bulbar muscular atrophy, and then did a postdoctoral research where she actually worked on a model of disease that you may or may not have heard of, C. elegans, which is a tiny microscopic worm, and she used that to study ALS. And I think that paper is just coming out. Is that true? Okay, good. So hopefully we'll get to hear, I don't know if she's going to tell us about her research background or not. So I know she has a research background, and it's great to have her as part of the association. So I'm going to let her give her talk, and then you guys will see me in a couple of minutes. All right? Thanks, Jill.
Thank you, Greg. Thank you, Maura and Amber. Okay, it's okay. Hold on. It always happens. Here we go. Steal. All right, we're in. Excellent. Thank you again for having me. So I'm Jill Yursak. I'm the Director of Mission Strategy at the ALS Association, and I have the pleasure to work with Maura and Amber the last couple months. I'm happy to be here. I actually, when I did my postdoc at Brown, we lived in Rhode Island for 3 years, so I'm always happy to come back to New England. So when I got the invitation, I was like, absolutely, I'm coming back. I live in New Jersey now. It's not as nice, so unfortunately, but that's where I'm from. Okay, so today I'm going to give a very quick overview. I only have a few minutes about our research program and all the great research that we're able to fund through the ALS Association. And then I have the pleasure to kind of be the MC for our research panel and introduce everyone up here today. So, all right, let's get started. It's thinking. Hold on. Maybe I'm doing the wrong one. Oh, we're in. Okay. Okay, so maybe not. All right, it's staying.
Nobody move.
No one move. Right. Thank you, Greg. Okay, so at the ALS Association, we like to say the world is our lab. And the reason why is because we fund research projects all over the world, not just one research lab. And so right now, we currently fund 115 grants. Throughout the given year, we usually fund around 150 to 175 grants that kind of cycle in and out during the year because they start and end. And this year, we're happy to announce that this actually just is very new news, is that we were able to fund 56 new grants in 2018 through our grant program. And we're really excited about that. And so we are the largest funder of ALS research, other than the National Institutes of Health. Our current budget is $18 million, and that is expanded since the ALS Bucket Challenge, where we had an average research budget around $2 to $3 million. So that's just very exciting. So what I want to talk about today, and I'll keep talking about, is that we can't do what we do without our partners and our collaborations that we have. And so we partner with academic scientists, like here people on the panel, and our clinicians. And we have industry partners and government partners like Paul at the CDC, that we all work together really to move the needle forward as fast as possible for everyone living with ALS and their families. So what we do at the ALS Association, and I don't have a lot of time, is that we fund research all across the research pipeline. And what I mean by that is that we fund research at the very basic level, from gene discovery and understanding what are those disease pathways that cause ALS at the cellular and motor neuron level. And then we also fund translational research, which is the drug development. So once we have a target, then we can develop a drug. Also, biomarker discovery, which is a way to understand whether a drug is hitting its target. And I'll talk a little bit more about biomarkers later and explain that more. Also, we do fund clinical trials. We currently fund up to phase 2 clinical trials. The reason why we don't fund phase 3 clinical trials is it's super expensive. And if we funded phase 3 trials, we wouldn't have any money really to fund anything else, just to be completely honest. We also, and I think this is one of the most important things that we do, is fund things that champion patient care through research. And what I mean by that is research that really helps people living with ALS more immediately. Things like assistive technology, helping people living with ALS communicate better. And also focusing on patient and caregiver care. How can we help you live better lives every day and decrease the burden that comes along with this awful disease? So today I want to emphasize that really pace of discovery has been on the rise, especially with the advent of it was like hitting the lottery with the ALS Ice Bucket Challenge. Since that, it's almost 5 years now, next August, there's been so much research that's been able to get done just because that huge influx of money, not only to us, but to ALS researchers really all over the world and other organizations. So one of the first examples I want to talk about is the ALS gene discovery. So part of ALS, we know, is inherited or familial ALS. That's about 10%. The other part of ALS is sporadic, meaning we really don't know what causes your ALS. But we do know is that there's been a lot of gene discoveries since the first gene discovery, which is SOD1 in 1993. And since the ALS Ice Bucket Challenge, we've had 5 new gene discoveries. This is the highest rate of gene discovery in the history of ALS, and we're so excited about this pace of discovery. So you can see in the top right, those top 5 genes have been discovered since the Ice Bucket Challenge. And what I want to emphasize is that people like Dr. John Landers at UMass Medical School, people at NIH like Brian Traynor, we have people all over the country and all over the world have been working together in really large collaborative gene sequencing projects. The KIF5A discovery, that involved 250 researchers all around the world. And a lot of those researchers were involved in these large collaborative projects like Answer ALS, Project Mind, the New York Genome Center, GTAC, CREATE. The list goes on. And we are able, through Ice Bucket Challenge, to fund all those huge projects to help move these projects forward. So it's a huge collaborative effort. Everyone's talking to each other. It's a beautiful thing. Researchers talking to each other. And that's something, it's common in ALS, because they know, and they can attest to this when they get up themselves, is that this is awful. This is an awful disease. We want to do as much as possible to collaborate and move things forward quickly. And we're really proud of that. And what's important about gene discovery is that each thing that we discover represents a possible new therapeutic target. So the more genes we discover are the more therapeutic targets that we can eventually, hopefully, get into clinical trials and then to you. So one of the other things I want to talk about today is the innovation that's happening in ALS. And one of the best examples of this is the ALS Assistive Technology Challenge. We did this challenge a couple years ago with one of our partners, Prize for Life, another ALS organization. And the goal of this project, which was about a year in the making, was to develop communication tools for people living with ALS to help them communicate with their families and loved ones and their doctors and everyone else as fast as possible and with as much ease as possible. And so we came together at our international ALS meeting, and this is actually in Dublin, and it was a really fun day. It was actually like, if you can think about Shark Tank and speed dating, it was kind of like that. They had 10 minutes, our 5 finalists, to talk about their prototypes, their great, their ideas, and sell their business plans and say, this is what we're doing, this is why it's awesome. So in the top left is our esteemed judges, and we had people from Microsoft, we had some entrepreneurs, we had people from some academic scientists come together, and they were in charge to figure out what was best. And at the end of the day, not everyone can win, right? But everyone came away with new partners and new ideas, and it was a really fun collaborative day. And so our second place winner was Python Technology. And I love talking about this group. So on the bottom left, in the middle, that's Dexter, and on the right is David, his partner. So Dexter has a really interesting story, which I'd like to really quickly share, because I don't have a lot of time. So Dexter's mom died of ALS. And during her journey, it became very frustrating to him that she couldn't communicate well, especially towards the end. And so what he did when she passed away, he took charge. He quit his job in Chicago in finance, moved to Massachusetts to MIT, and started this company called Python Technology, which is actually named after her cat, who she loved, Python. And so he hired his friend David Chipolletta, who was an underwater robotics engineer. So you can imagine how smart he is, right? And so they came together to make this technology. It's called a wearable muscle sensor. And what it does, and it's morphed since this technology challenge in the last 2 years, it's actually now a watch, and it can be moved around. And it sticks onto you, or is a watch, and it actually detects the smallest muscle twitches. And those muscle twitches can then be translated to help you type on the computer, they're working on things to move your wheelchair, things to control things in your home. It's really, really cool. So we're so excited to see him and his project move forward. Since we gave him $100,000, he's raised over $7 million for his company, just with our seed money. So we're so excited. So this is a great example of how we've been able to maximize investments through a really great idea. Lastly is biomarker discovery is growing, and biomarkers are really exciting too, because it's a way to understand whether a drug is hitting its target. So pharmaceutical companies are looking for biomarkers. And so if they know if their drug is hitting its target, they're more likely to invest in ALS, and that's what we want, all this investment in ALS. And biomarkers really can track disease over time and also help with diagnosis, because right now diagnosis takes about a year, and that's too long. So this would be a game changer. And so we have, and I don't have time to talk about it, but many biomarkers in the pipeline that are very, very close to coming to market that will really help with diagnosis and understanding disease progression. And biomarkers, just so you know, and I don't have a lot of time, are a way to track something in your body. So if you think about it in the autumn, the fall, you have leaves that change color over time. It's a biomarker, so something you can track over time. But in your body, it's a marker that's in your blood, something you measure in your cerebral spinal fluid, or even your urine. So it's a way to track something and how your disease is progressing over time. Okay, and so we also have increased numbers of enrolling ALS clinical trials. I did a search with Niels, which is one of our great partners, and Suma, she may be talking about that today, is that right now we have, I'm done, looks like, so we have 32 trials, well, 63 total. So I'll let her talk about this today. But I do want to say that we funded so many of these trials, either at the preclinical level, at the clinical level. And we have growing numbers of association clinical trial sites all over the country. And this is our most current map. And there's 4 here in New England, and we're hoping to expand in the future. So this is something that I really wanted to also touch on. So that just totally died, so that's it. So I want to say that we do have partners that are keys to our success, and my slides will be available in PDF form. I'll give them to the chapter, and there's some really great resources at the end that I've added, so you have them for you in the future. So thank you. It was really quick, and I'm sorry I talked fast. I did my best in 10 minutes. So thank you so much. And now I'm going to introduce again Dr. Gregory Cox. So I'm not going to do an introduction again, but we are proud. We have funded Dr. Cox in the past, and his mouse models are really, really important to understanding disease pathways and discovering new disease pathways in ALS, which also represent new therapeutic targets. So Greg, I hand it over to you. Thank you.
All right. I'm not going to show any slides. We have such a short time, and I'm the odd guy out here because I'm the mouse guy. So we heard about how they fund basic research, and that's really who I am and what we do. So we're the end of the research that the patients never really see. So that's why this is so important for me, because I study ALS at the molecular, cellular, and animal model level. And so I'm going to talk just a little bit about the animal models and how we use them as a preclinical model to understand the disease, but also to test therapies. All these therapies that are coming into clinic had to be tested somewhere. They can't be tested on patients because we don't know if they're just going to hurt you or if they'll have no effect at all. So they have to be tested somewhere. And they're tested on our mice. And so how do we end up with a mouse that's useful for an ALS patient? The tricky part is, and we saw the number of genes that have been discovered, ALS isn't one disease. I mean, it's a constellation of symptoms that we call ALS, but there's not one cause of ALS. Some genetic diseases, there's a single gene that explains every patient out there in the world. That's not true for ALS, unfortunately. So that list of all those genes each explains a small percentage of the patients as to what's causing their disease in a familial case. So what do I do with these mouse models, and how do I generate these mouse models? So in some cases, the gene discoveries that we just heard about, these amazing discoveries, we can actually take those discoveries and make a model for the disease. Because our research mice, we can do some really cool tricks with our mice. We can take these mice and we can change their genome, we can add genes to them, mutate genes, make changes to their genes, just based on what we find in patients. And we can put exactly the same mutations into a mouse. And then that mouse will go on to develop ALS. And so we've done that for a number of cases where we've had discoveries in clinical trials and sequences, and we've made those mutations in our mice, and our mice now develop ALS. So now we have a reproducible model that can be tested for drugs and tested for understanding the mechanisms. We all want to understand why ALS happens. And given that there's lots of different genes that account for 10% of the patients, we really want to know what's contributing to the rest of the 90% of ALS patients. That's what we really want to understand. So we can do these cool tricks in our mice, make models that can then be tested for all the therapies that are hopefully going to come to help you. But what do we do about the 90% of patients where we have no clue what the cause is? That's the hard part. So one of the coolest things, so I'm at the Jackson Laboratory in Bar Harbor, Maine. So we're on an island on the coast of Maine. We're a nonprofit research institute, a small place. But there's a couple of us there at the lab that we work on neuromuscular diseases. But also part of the Jackson Laboratory is they produce lots of research mice for all the researchers around the world. I think they shipped over 3.5 million mice last year to researchers around the world. That's a lot of mice. And in the process of breeding all those mice, because mice are bred, they don't just make them on an assembly line, so they have to breed these mice. And so there's lots of mouse breeding going on, and Spontaneous mutations are really rare, but if you breed 3.5 million mice a year, these spontaneous mutations show up, and they show up so frequently that every 2 weeks, we have what's called the deviant search. Funny name, it's a deviant search, and they announce it over the PA that the deviant search is happening. And so what is the deviant search? So the deviant search is the animal caretakers at the Jackson Laboratory who are breeding all these mice and changing the boxes and feeding them. They notice when something is wrong, because with our strains of mice, we have all these inbred strains of mice. Every mouse within a strain is identical, and that's an amazing tool that we have for mouse genetics. I can work on a mouse in Bar Harbor, scientists at MIT can work on that mouse, in Europe can work on that mouse, in Japan can work on that mouse, and we're working on exactly the same mouse because they're all identical. They're like a whole room full of twins. And so it's a powerful trick that we have with mice because we have these completely inbred strains. But when these animal caretakers are changing the mice and somebody looks different, it stands out because everybody's identical. And then all of a sudden, one has a different coat color, or one walks funny, or one becomes thin, wasted, and paralyzed. Those are the mice I like. I'm a really sad person. I go to the deviant search looking for mice that are paralyzed. But why do I do that? Because ALS, and because of other motor neuron diseases and muscular dystrophies that affect motor function. That's what the symptoms that you experience. The mice experience exactly the same thing. They have spinal cord, motor neurons, muscles. They're exactly like us from a neuromuscular standpoint. So when they inherit a new mutation that causes them to become paralyzed, I get to take these mice and bring them back to my research lab. So I love the deviant search. And so when I get these mice from the deviant search, they have the disease that I'm interested in. The mice have a progressive paralysis phenotype. And it's my job, and the students in my lab, we study these mice, and often we'll breed the mice if we can, or we will do in vitro fertilization if they don't breed. We can do some special tricks with our mice. But if we can propagate this line and reproducibly generate mice with this disease over and over again, we know it's an inherited cause of the disease. And then we will do genetic positional cloning. We'll identify the gene and the mutation that causes it, and we'll add to that list of genes that are discovered that can cause ALS. And we've done that a number of times for different neuromuscular diseases, where a funny sick mouse that showed up in our deviant search is now a new model for a motor neuron disease that didn't exist before. And so we can use these now to understand what causes ALS, what causes motor neurons to become sick. Because we have these mice that we can breed and make many of, we can put them into different treatment groups, give one a drug, give another just a placebo, and we can determine whether or not treatments will help. So from a basic science standpoint, we're discovering genes, discovering mechanisms that cause motor neurons to become sick and die, but we're also generating models that can be used by everyone around the world, because once we discover them and describe them, then we give them away to people all around the world, drug companies, other researchers. Our job is to provide models for everybody so that they can test the latest and greatest drugs. And as we know, most drugs fail, so you want to fail quickly and cheaply so that you can get to the ones that work. And so by providing these models to researchers around the world, that's what's going to push drugs into the pipeline. And so I will end here just saying that our work at JAX and the students and postdocs in my lab, we love to be connected with patients and their families because we are so removed. My patients are small and furry, so I love coming here and getting to meet patients and their families because that connects me to the diseases that I'm most interested in studying. So I will end right there, and thank you guys very much.
Thank you, Greg. That was great. So next we have, let me just get my bios, Dr. Suma Babu. Suma is a board-certified neurologist and neuromuscular specialist at Massachusetts General Hospital, or MGH. And an instructor at Harvard Medical School. So she currently is working to develop innovative molecular outcome measures to determine target engagement and dose selection in early phase 2 clinical trials in ALS. So Suma is really heavily involved in the clinical trial space at the Northeast ALS Consortium and at MGH, which is a huge and wonderful program that runs, you said at least 10 clinical trials you're running right now, hoping to run. It's a big lift, and Suma is a wonderful person, and we're really excited to have her today and to talk to you about her research updates. So thank you, Suma.
Thank you, Jill, and thank you, ALS Association, for having me here today. I'm going to quickly present some research updates that's going on at Mass General Hospital, and no disclosures relevant for this talk, and I have received research funding from ANEM and Orion Pharma, and I'm currently an investigator on all the trials that are listed on this slide. A little bit about me. I grew up in a city from Bangalore, called Bangalore in India, which is now known as Silicon Valley of India. That's a picture of my medical school, and then I came to Washington, D.C. area to do my master's in public health at the University of Maryland. Moved on to Cleveland Clinic to do my neurology residency, and then came to Mass General and Brigham and Women's Hospital to do my fellowship in neuromuscular and a separate fellowship in ALS, and then stayed on as faculty. And a big thank you to my chief of neurology, Dr. Merit Sukovich, who is truly an inspirational leader in ALS research, and also acknowledging more than 100 of my colleagues who are working very hard every day within the Mass General ALS team to ultimately find a cure for ALS. And there are lots of really exciting developments happening in ALS all around, from clinical care to gene discoveries to treatment trials to biomarker discoveries. And I'd like to focus on 3 important messages today from Mass General. One is about early genetic testing. Jill touched upon the value of genetic discoveries and the role of treatment trials for gene-positive patients, as well as the role of brain PET imaging as a biomarker for detecting inflammation in ALS. There are multiple trials that are ongoing, currently enrolling in the pipeline at Mass General for ALS, and I want to talk about one of the trials, levosimendan, today. And so this is a familiar slide for you already. There's an exponential growth in the gene discoveries recently, and most recently we've had a gene discovered called the KIF5A gene, and as Jill mentioned earlier, a huge thanks to all the patients and families who have participated in these large biomarker studies, including ANSWER ALS. Mass General is one of the sites for ANSWER ALS that has led to this discovery. And the question then arises, is genetic testing applicable for me? Should I consider genetic testing? We're learning more and more that there are multiple different phenotypes where diseases may not really pass on from one generation to another generation. Multiple of these gene mutations that cause ALS may actually skip generations, and they may not be a known family history of ALS. But there are also genes that cause ALS as well as a different phenotype called the frontotemporal dementia, and within the same gene mutation with multiple different family members carrying that gene mutation. So if there's any family history of ALS or dementia, definitely talk to your ALS physician to see if you might be a candidate for genetic testing. It's more important now because there are more and more treatments coming through the pipeline that are targeting gene-positive ALS. There are multiple ways to get genetic testing done. You can get that done through CLIA-certified laboratory, through the multiple laboratories that do genetic panels. But one issue we run into is that insurance companies often don't pay for these genetic panels. NEALS, the Northeast ALS Consortium, has a brand new protocol that is funded by ALS Association as well as MDA that offers genetic testing for patients with ALS that are seeking clinical care at a NEALS site, and this would come free of cost. It's not available at this moment, but we're hoping for it to be up and running in 2019. So that's an option you have. And then that's Dr. Katie Nicholson, who's my colleague at Mass General, who is leading a wonderful effort called DIALS, and this is Dominant Inherited ALS program. And through this program, you can get genetic testing for asymptomatic family members who have a family member with a gene-positive ALS. And a similar effort is ongoing at Miami by Dr. Michael Benatar as well. So these are different options we have for genetic testing. And this is really important because there are more and more treatments coming through the pipeline. We have 3 current ongoing trials for gene-positive patients, and 2 of them are run out of Biogen. It is using a technology called antisense. And another really exciting trial is using a viral vector, and that's very early on, and 2 patients have been dosed with this medication as of now. So the hope is that with successes of these programs, there will be a day we'll be talking about preventive trials to prevent symptom onset if we know that somebody's carrying a gene mutation. And that's kind of the hope for the future as well. In terms of biomarkers, our group at Mass General that's led by Dr. Nazem Atassi, we have an imaging core, and we are looking to see what is the role of PET imaging of the brain to identify inflammation, to identify various different possible pathophysiological mechanisms in ALS. Earlier, the thought was all ALS is one disease, but now we are starting to learn that ALS is not all one disease, but there are many different mechanisms that's driving the disease in different patients. It could be inflammation, it could be something called glutamate excitotoxicity, it could be something called epigenetic changes, and so on and so forth. We're using multiple different PET tracers targeting each of these pathways to see if we can characterize and quantify these changes in the brains of people with ALS. And so far, we've been very successful with this PET tracer called PBR28. It is a marker for inflammation, and we have scanned about 75 patients with ALS, and the picture on the right that has an orange signal is the area where inflammation is detected in the brains of people with ALS. And if you look at the neuroanatomy of the brain, that area corresponds to the motor cortex. And motor cortex is like the headquarters for controlling muscle function, the arms, legs, speech, and swallowing. And the picture below has a little blue dot corresponding to the same region where the orange is, and that's the same area of the brain that shows a little bit of cortical thinning where the inflammation is occurring. And this is a very exciting, very promising biomarker, and currently we're using this biomarker as a clinical trial outcome in at least 4 clinical trials. And the idea here is that if this is successful, it will really shorten the duration of clinical trial testing, especially in early phase 2 trials. And as Jill had mentioned earlier, it's very quick to identify whether a drug works or not, and Dr. Cox had alluded to that as well. If a drug doesn't work, let's not take that drug forward for further testing, and that can be detected soon, very early on with smaller sample sizes as well. And inspired by the success that we've had with the brain imaging, now we're expanding this to spinal cord imaging as well. We recognize and acknowledge that everybody's ALS is not the same. Some people have more of the upper motor neuron burden, and others have more of the lower motor neuron findings. So people who do not have those brisk reflexes suggestive of upper motor neuron burden, we're trying to see if we can identify similar patterns of inflammation in spinal cord as well. And it's all very early, and we're seeing some good early signals of thinning of the spinal cord in patients with ALS. And in terms of trials, there are multiple trials ongoing right now, and there's an explosion of interest in developing drugs for ALS. There are at least 15 active phase 1 trials, about 30 phase 2 trials, and more than 80 companies that are investing in ALS drug development. And this is a list of all the trials that's ongoing at Mass General. I have brought some brochures if you'd like to take home any of the trials. It's out of at the Mass General desk up front, outside this room. And one trial that's ongoing right now is the levosimendan trial. It's a very unique trial. It's a phase 3 trial that's ongoing both at US as well as in Europe, in about 101 different sites, and 450 participants are being enrolled for this trial. Unlike many of the trials that you probably have heard of that are targeting disease progression slowing, this is a unique compound that's actually not working at the level of the brain or the spinal cord. It's actually working at the level of the muscle downstream. And this is a cardiac failure medication that improves the muscle force of the heart, and heart is a muscle, and the thought here is if it can improve the muscle force of the heart, can it also improve the muscle force of skeletal muscle and improve function in patients with ALS. They're using diaphragm as a readout for this trial, so people who have a little bit of respiratory difficulties with a vital capacity between 60 to 90 will be eligible for this trial, and Mass General is one of the sites for this trial. And Centaur Amylyx is another very exciting trial that is led by my colleague, Dr. Sabrina Paganoni. And this is a combination of TUDCA and phenylbutyrate, targeting the motor neurons and targeting the oxidative stress patterns occurring in motor neurons that might impair their survival. This trial is using multiple different biomarkers as outcome in addition to the ALSFRS-R, including the brain PET imaging, as well as blood biomarkers and neurofilament level and quantitative strength testing as well. And the stem cell trial, Neuron by Brainstorm, is another very exciting trial that's ongoing at Mass General and the other sites that are listed there. And this is taking the patients. Thank you. I think I'm out of my time. And this is some information if you want to contact our team for any of the research studies. Thank you so much.
Thank you, Suma. So next on our agenda is Dr. Elijah Stomel. So Dr. Stomel did his MD at Boston University in Boston and his PhD at Boston University School of Medicine, and is currently
Page 2.
What?
Page 2.
Well, yeah, he's a PhD. Yeah. But anyway, so I'm just looking here. Oh, here we go. He is currently professor of neurology at the Geisel School of Medicine at Dartmouth, Hanover, and New Hampshire, so that's very close by here. And Dr. Stomel does a lot of great work in the environmental realm of ALS research. We've been able to fund him for many years, and we're really proud of that. So thank you, Dr. Stomel. I'll hand it over to you.
Good morning. I don't know how my voice is going to work here. I got one day of the year where I get something like this. I recognize many of you out there, and thank you for coming. So the arrow will take us ahead, I think. So ALS is, as already mentioned, is not one disease, but several diseases. And it has similarities to other neurodegenerative diseases like Alzheimer's disease. Thank you. Parkinson's disease, frontotemporal dementia, and even multiple sclerosis. I don't know what's wrong with this thing. Okay. And all these disorders most likely have a genetic component where there's a predisposition to developing the disease, and on top of that, there is some environmental trigger for the disease. So with the help of our ALS patients, their families, and with research going on around the world, I'm optimistic that we're going to find good treatments for this sort of array of diseases that fall under the realm of motor neuron disease in the relatively near future. And I'm indebted to the National ALS Registry, the CDC, and the ALS Association for funding and for help. This is I don't think I'm going to be able to make this work, but this is a motor neuron that we cultured from a rat spinal cord. And if the cell body that sits in the spinal cord were the size of this room, the axon that goes out to your muscles would probably go all the way to New York City. That's how big these cells are. They're huge. I can't make that work, but it's a nice time lapse that shows the particles moving up and down the axon. And those particles, when they move at about 1 micron a second, will take up to 2 weeks to go from your spinal cord down to your feet. So they're huge cells. All of these things listed here are considered to be risk factors for ALS, and I know we're very pressed for time here. And we've been looking at things like heavy metals and cyanobacteria, and I'll go into that in more detail. You've seen the slide about 3 or 4 times now. The idea of the take-home message from this is not only that there's been a lot of work looking at genetic risk factors for ALS, but also the fact that there are many, many causes for ALS, many things that can go wrong. And for one person who has ALS, it can be a totally different issue in somebody else. The whole field of epigenetics is important in ALS, and what genes are turned on or off, and the environment likely turns genes on and off. If you look at 2 chromosomes from a 3-year-old set of twins on the left, they look pretty much the same. And when you get out to about age 50, the genes that are turned on or off are quite different. And these are just typical pictures of twins that have developed over the years characteristics that don't look the same as when they were younger because they're not exposed to the same environmental issues. This is Keith Richards when he was about 18 on the left, and this is him at about age 55 on the right. And at age 73, that's his twin brother on the left, and that's Keith Richards on the right. So the environment has an influence on how you end up looking and what happens to your body. He didn't have a twin brother. I think that was Prince Charles on the left. Although maybe they're related. So when you think of ALS when it was first described 200 years ago or so, the environmental risks have probably changed dramatically. And so, it's like resetting the clock. Each decade there's something new in the environment that we're exposed to and could potentially be important. So it's a very hard thing, and then you have this lag period where you might have been exposed to something as early as in utero, and then you don't even know or remember what you were exposed to 30 years ago. In Australia, these are death records there, the mortality rate has doubled in the last 20 years in Australia. Nobody knows why. On the left are some early studies looking at gastrointestinal cancers on big databases. This was done back in the 50s. And they looked at the log of the age of onset versus the log of the incidence of disease. And you have these slopes that look very characteristic to what you see on the right, which is a big group of ALS patients. And I'm not going to get into the math, but these slopes and these curves suggest that there are great similarities between cancer and ALS. We all know that there are environmental risk factors for cancer, such as smoking and other many myriads of things. In Guam, back in the 1940s, there was a rate of ALS and some other diseases like Parkinson's disease and a dementing illness that was 200 times higher than the rest of the world. And it was thought to be the Rosetta Stone. People went there and looked at what people were eating there. They were in sort of a starvation mode. And it turned out that in the roots of these plants were cyanobacteria. That was a cycad plant, and they were making a flour out of it. And in that flour, they found a toxin called BMAA, which is actually an amino acid made by cyanobacteria. And it actually will fool the translational process in cells and get into cells and make the proteins misfold. And it will also bind to certain receptors, which are neurotoxic. And BMAA is on the right there. There's another disease called latherism, which is a form of motor neuron disease, and you can see how similar those two toxins are. And the one for latherism comes from cycad, I'm blocking it a second, from a...
Cycad seed?
No, the cycad is on the right. Anyway, it doesn't matter. We're so rushed here. So, But anyway, they were eating this vegetable that had this toxin in it. In Enfield, New Hampshire, we saw clustering of ALS. This is about 9 or 10 years ago. We didn't know why, but it turned out that the same cycad, the same cyanobacteria that were in the cycad seeds were in this lake, and they had annual events there of cyanobacterial blooms. Paul Cox and his group fed monkeys the BMAA, which comes from the cyanobacteria. He was able to reproduce both Alzheimer's disease histology and the Guamanian-looking ALS by feeding the monkeys BMAA for about a half a year. And he was able to block that by using the amino acid L-serine, which is the amino acid that BMAA substitutes for in the translational process. Those are just some pictures. These are of what the brain tissue looks like. Both, it looks like Alzheimer's and Guamanian ALS. These algal blooms or cyanobacterial blooms are ubiquitous. They're found all over the world, and they're probably even found in cooling towers on building roofs. This is northern Vermont on the Lake Champlain. This is in Florida, which has had a terrible problem. This is in the Baltic Sea, and they use shellfish there to try to clean up the lakes, the sea rather, and then they dump the shellfish on farm fields, so they're actually putting the toxins onto the farm fields. They use it as a fertilizer. In southern France, they describe clusters of ALS, and they grow oysters there in these lagoons, the Thai lagoon. And here we're looking at two maps, one in northern Lake Champlain and one near Tilton, New Hampshire. I've seen conjugal couples, people living in the same house where they have these blooms come down with ALS around the same time. These are some of the clustering of ALS along Lake Champlain, where there are big blooms every year. This is a study we did looking at lakes, and we made an association between the cyanobacterial blooms and ALS. We set up these aerosol collectors, and we can find, because they have a pigment in them, we can actually see the cyanobacteria light up on the filter papers after we've collected air samples for a while. And we've looked at post-mortem cases and been able to find it in the lungs of patients. Just any old patient that comes to post-mortem, it has an autopsy done, and it turns out that the patients that have the cyanobacteria in their lungs have a much higher rate of neurodegeneration. We've been able to find, with bronchoscopy, just patients coming for workup for lung disease, we've been able to find cyanobacteria in almost all comers who come to the lab if we use PCR, which is another technique. So we've also looked at ALS patients and methylmercury, which is a neurotoxin, and we've been able to show that ALS patients tend to eat fish that are high in methylmercury, like swordfish and tuna fish. And they also have higher levels of methylmercury in their toenails. We have a big biorepository given to us by the ALS registry of nail samples that we're going to repeat this study on. And we're looking in many states now, Ohio, Florida, and northern New England, and trying to look at all the potential toxins that are out there. This is Lake Erie in Ohio. We have a new grant where we're going to work with the NIH and Brian Traynor and try to look at genetic information on patients that we're collecting and compare that with our questionnaire data and our maps. And we have some funding through the ALS Association to look at aerosol collection. So we have 2 clinical trials. If anybody knows anybody who wants to be in a clinical trial, we have 2 going on at Dartmouth now. The plasma exchange trial is basically winding down now, but the L-serine trial is just starting. L-serine has many mechanisms of action. So I think I'll have to stop there. Sorry about my voice. Boy, that was rushed.
Thank you so much. I hope you feel better. So last but not least, we have Dr. Paul Maida. So Paul comes from us is a principal investigator at the United States congressionally mandated ALS National Registry and is responsible for providing medical, scientific, and epidemiological expertise on matters related to ALS. So last few years working at the ALS Association, I've been a pleasure to get to know Dr. Maida, and we've been a partner with them, I think since 2008, since the registry has been founded. So a really great partnership, and we're happy to have him today to talk more about the ALS registry and the biorepository that that Dr. Stomo talked about today. So thank you, Paul. There you go.
Thank you, Jill, and thank you, Amber, and thank you, Mark, for the opportunity to come to New Hampshire to give this talk. Before I get started, I don't know too much about New Hampshire. I do know every 4 years, a lot of people come here, politicians and so forth, and you guys have the best license plate slogan in the country: "Live free or die." That's for sure. So let me go ahead and get started. No disclosures. Brief outline of the presentation right here. How can you join the fight against ALS? Why is the registry important? Why is the biorepository important? And what do we do? How can you join? And how can you join clinical trials and epi studies that we help to recruit for? And we have risk factor surveys. Why go ahead and take those surveys? And what do we do? So the National ALS Registry, we look for what causes ALS. For us, it's important to solve the mysteries behind ALS. So we fund research, we publish research, and so forth. I've got a couple of videos, which I'm not going to show you, but if you could please go to our website, cdc.gov/ALS. You can see these informational videos about the registry. And any questions I'll take later. But very quickly, why is the National ALS Registry important? Why should you join? So like I mentioned, there are more unknowns and knowns about ALS. We're not sure what causes 90% of ALS. So the registry's goals, it's pretty much to seek what causes ALS, the etiology of ALS, seek to find out the risk factors, environmental, genetic, occupational behind ALS, as well as to see who gets ALS in the U.S., report the number of cases, which is incidence as well as prevalence. This is your registry. It's mandated by Congress. It's strictly voluntary, but this is, like I said, this is your registry. I'm a caretaker, and this is for you to kind of pretty much join the fight against ALS. You know, we like to say ALS research counts on you. And the National ALS Biorepository, you know, this is part of the registry as well, and this allows us to look for areas, for example, in genetics, identification of certain genes, as well as biomarker identification. Currently, there is no blood test for ALS. We cannot go ahead and, you know, prick someone's finger and say, well, hey, you've got ALS. We have to go ahead and look for certain biomarkers out there. So the biorepository is looking for these biomarkers, as well as disease progression, as well as looking at if certain drugs will work against ALS or not work. We're also looking at environmental risk factors, heavy metals, lead, arsenic, and so forth, as well as persistent organic pollutants, for example, exposures to pesticides, insecticides, and so forth. So details of the National ALS Biorepository. Our biorepository, like I mentioned, is part of the registry. When you come to the registry and you join, there's a question, do you want information on the biorepository? And if you click yes, you fill in your information, and we'll go ahead and mail you a packet on the biorepository, and we'll contact you in about a week or two later to see if you've got this packet and if you have any questions. This biorepository is very unique. It links the risk factor survey data, which I'll go over in a couple slides later, with the biosamples. It's nationally representative, so it's beyond a certain referral center. We go coast to coast, and we go to your home. It's user-friendly. There's no charge to you whatsoever for us to come to your home to go ahead and collect your blood, urine, or saliva. And we also have a post-mortem component as well. But these are also pristine samples as well. So these are samples which are not being left over for researchers from a current study or something like that. These are pristine samples available for researchers. And our goals for FY19, we want to collect 325 bloods, 125 salivas, and 10 post-mortem as well. And like I mentioned, this is probably going to be the largest biorepository in the world of pristine ALS samples in about 3 to 5 years, we feel. This is a very quick schematic of the biorepository. We have the in-home collection, blood, urine, saliva. We have the post-mortem collection that kind of comes together in the biorepository. The risk factor surveys are taken when you join the registry, and the specimens are then sent out to researchers to go ahead and solve the mysteries behind ALS. We also have these clinical trials. So, I mean, there's a whole bunch of clinical trials that were listed in the slides from our colleagues right here. So the registry helps you recruit for these clinical trials. When you join the registry, you can say, I want to be notified about clinical trials. We send an email to you when a clinical trial is available. And so we never disclose your information. No patient information name is ever disclosed. So we simply send you an email with a link to the clinical trial itself. And to date, we have over 35 institutions who've actually used this system for clinical trials. And these are some of the notable clinical trials, many of them which were mentioned by our colleagues here, the one for Ryan Pharma, the one for Amylyx Pharmaceuticals as well. Cytokinetics did one for 42-day ALS, Flex Pharma, FLX787, Neuraltis, as well as some other ones as well. So we work with pharmaceutical companies to go ahead and help them recruit for their clinical trials. So we also have risk factor surveys that we encourage people to take. And you're asking me, well, why are these important? Well, these risk factor surveys helps us, gives researchers more clues about ALS. So for example, certain occupations are at higher risk for ALS than other occupations. Military service. Veterans are at greater risk for ALS than non-vets, so we're not sure why veterans are at a higher risk for ALS. Physical activity, smoking, drinking, are these risk factors? We know smoking and alcohol appear to be a risk factor for ALS as well as physical activity. For example, insecticide exposure, pesticides, where did you live? Were there certain areas where they were spraying pesticides or insecticides? Were these possible chemicals linked to ALS or not? Traumatic brain injuries, did you have a concussion or a TBI when you were younger? So these are all these questions we ask patients when they join the registry. We currently have 17 risk factor surveys, and they can be taken in any order and at leisure. There's no reason to take all 17 at one time. And so, but these surveys are very important for you to take and complete so we can go ahead and solve the mysteries behind ALS. This right here is a quick breakdown of the surveys we have. We have demographics, occupational history, and so forth. We also have a survey on ALS-FRS, which is a longitudinal survey where we ask you when you first join, how are you progressing? And every 3 months we send you an email saying, gauging your progress. We also have a unique survey, which is an open-ended survey, which allows patients to tell us in their own words what they think may have caused their disease. So to date, we have almost 80,000 completed surveys, and all these surveys are also linked up with the biorepository samples. So we have a great rich information of patients' occupation. So for example, let's say we have a patient who was a plumber who was in the military and they have ALS, so we have all that rich data sitting with us and we're actually currently analyzing. So very quickly, how many cases of ALS are there in the U.S. right now? So very quickly, let me just go over. ALS is not a reportable disease or a notifiable disease. In the area of public health, if someone gets the flu or has got tuberculosis, that information is sent to CDC. ALS does not work that way. We have to go out and figure out and do case ascertainment to see how we go ahead and figure out who's got ALS. And we use national admin databases like Medicare, like the VA system, to go ahead and do case ascertainment. We figure we capture about between 80 and 85% of all ALS cases in the U.S. And we can all agree one case of ALS is one too many. That's why the National ALS Registry exists, to go ahead and solve the mysteries behind ALS. We just released a report last week, and so currently we have about over 16,000 cases of ALS, which is about 5.2 cases per 100,000. ALS continues to be more common in whites, males, and persons between the ages of 60 and 69. We're not sure why males get ALS more so than females. So we also fund research as well. Research is very, very important. People are like, hey, what's happening in research? And so the research we fund, we just picked up 3 new grants back in September to go ahead and glean more information about the causes and the risk factors for ALS. Let me just quickly go over some of the areas we're funding. We're privileged to fund Dr. Stammel here, who's helping to identify cyanobacteria and those individuals living close to large water bodies, and if they're potentially could be exposed to microcystines, cyanobacteria, which could potentially lead to ALS. He's helping to solve that mystery right there. We're also looking at pesticides and pollutants. We're looking at biomarkers and genes, as well as the role of hyperlipidemia, which is excess fat in your body, in your blood, and so forth. Autoimmune diseases, as well as biomarkers, as well as genetics as well. We're also looking at something called oxidative stress. So over time, your body builds free radicals, and these free radicals can cause harm to your body. So we want to figure out, why does oxidative stress, does it lead to ALS or not? So, and just in closing, I just want to say the National ALS Registry does a lot more than just count ALS cases. Like I mentioned, we do the biorepository. We have the epidemiology part, looking at who gets ALS, incidence, prevalence. We work with the ALS Association, which is one of our closest partners. We have the risk factor survey data, which we certainly would encourage you to go ahead and take and complete. Notification system, which allows patients to be connected with clinical trials and epi studies. And we also fund research. So, and the videos I mentioned, we can go ahead and if you could please go to our website. We also have a booth in the back. My colleague Tom Hicks is here. Myself can answer any questions if you have regarding the registry, how to enroll, how to reset your password, and so forth. Thank you.
All right, thank you, Paul. Okay, so we have a few minutes for questions, so we're happy to take some questions. Do we have a microphone? Are we going to take my microphone? Okay.
There's a lapel for you to use.
Sure. Sure thing.
That's roaming.
Okay, hold, we have our first question.
That's going to take too long.
Hi, my name is Bob. I want to say some things before I get to the questions that I want to ask. First of all, my background is from product management technology, so I am used to dealing with software development, hardware, software engineering, and representing what the market will bear and needs are, what customers want, number one. Number two, I've always found out that there's Y amount of money and Y amount of time, and there's X amount of things to do. X is always greater than Y. Third thing would be that, to honor Greg Cox, he said he's used to working with things that are small and fuzzy. I am neither small nor fuzzy. But why I ask the question is to help focus the effort that you're doing onto what we need. I lost my better half to ALS. I was the 7x24 caretaker and watched her slip away 1 week at a time over 20 months. She retired and we were going to play golf and travel. Instead, we went to hospitals, doctors, labs, MRIs, CAT scans, tests, and more tests for 9 months before we were given a diagnosis on July 25th, 1916, not that I remember the date. So 9 months to find out what we were dealing with. I hear everything you're talking about, but I'd like to use an analogy and get to specific questions. If you're doing a rowing race down in Cambridge on the Charles River, and you have 12 boats on the river, each boat is putting optimum output out, 12 different boats, and they're going to get to the finish line at different times. But if we wanted to optimize to get to the finish line the quickest, we'd put the 12 people in one boat. Same amount of effort, but it would get us to the finish line quicker. For those of us who are dealing with ALS, we want the solution quicker. Let me give you the objective we're going to. Just in today's environment, you can go with cancer. You can go for chemo and radiation and become cancer-free. What we want is treatment that you can go to ALS-free, just like cancer-free. And we can term an entirely new definition from ALS being amyotrophic lateral sclerosis to another loving situation that can live a normal life. How do we get everybody? Some comment that we're doing 31 projects. You think that's good? I'd rather have us working on 3 that it would get to the end quicker, because for my loss, I would have liked to have her continue her loving life and let us play golf. Is everybody on the same boat? And we're not confusing effort for progress, because what we want is a fix for ALS, and we want it sooner. Any comments?
Okay. Clinical, and say, I mean, I can, as I said at the beginning, if ALS was one disease, then everybody's focus would be on exactly the same project and problem. given that ALS has multitudes of causes, we saw the list of genes that are known, and that only accounts for 10%. 90%, we have no clue what's causing it. It could be some combination of the environmental. So, given that every patient is so different, if everybody worked on one tiny sliver of ALS, whether it was a SOD1, that's the one we've known about the longest. There are clinical trials underway for that one subtype right now that are likely to be very effective, and we can hear more about that. So when we have a focus, we can make decided progress exactly like you described. The problem is right now, ALS is a shotgun. All the patients are so different from each other. One therapy that would be awesome for your wife, would have been beneficial, would do nothing for 90% of ALS patients. And that's what's killing us right now. And it's finding out what the true causes are so that patients can be grouped effectively so that the clinical trials tell us the answers we want to know, yes or no, this is good. And so the quicker we get to that point, the quicker we will make the progress that you're demanding. And I'll let the clinical folks talk.
I'd like to add to what Dr. Cox said is you're absolutely right. We are all hoping to make that progress as quickly as possible towards finding a cure. Traditionally, a drug, when it moves through phase 1, phase 2, and phase 3 trial towards FDA approval, it takes about 10 years. And if you have 30 different compounds, and we need many patients to enroll in each of these trials, and it takes much longer to actually get one single drug approved. There's a lot of discussion in designing more efficient clinical trials. There are talks about newer designs like platform trials, where we cut down on the startup of each trial, where we're testing multiple drugs in tandem within one master protocol. That's being discussed, or even basket trials where we have multiple, as Dr. Stommel had mentioned, Alzheimer's, ALS, and the multiple neurodegenerative disorders are like cousins of each other. They're common pathways by which these disorders can morph into symptomatic phase. So if we can target a compound that might be effective not just in ALS, but in multiple different disorders, having similar common pathways, that's another faster way of moving things forward. And using biomarkers is going to be a huge way to actually help speed up the progress very quickly. The traditional way of assessing if a drug works or not is by using survival or ALSFRS-R, which we've learned that requires a lot more patients to be enrolled in a trial and for much longer durations. If we can use a blood biomarker or even imaging, that'll really cut short the duration of testing and take the drug to the next phase very quickly if it works.
In cancer, there already is this precision medicine where, depending on your genetic makeup and what's going wrong in your cancer cells, you can target multiple targets in a cell and get rid of it. And I think that eventually that same precision medicine will be used in diseases like ALS, where you'll be able to identify several targets. And then you'll have to have several medicines. As with cancer, you don't treat cancer with one medicine normally. And you'll be able to affect neuroinflammation, you'll be able to affect apoptosis, you'll be able to affect free radicals, reactive oxygen species. You might be able to turn off a gene, but it's not going to be just one therapy. But if you can identify sort of common denominators like neuroinflammation, we're doing this plasma exchange trial now, and that's supposed to reduce neuroinflammation, and it may have some benefit, but it's not going to completely cure the disease because there are other things going on at the same time. And to identify all those things that are going wrong and to then come up with ammunition for all those targets is how it's I'm sure that you'll be able to someday be able to cure all these varieties of ALS. And there's frankly very few clinical trials going on where an attempt is being made to use a cocktail of medicines, and they're just too expensive and there are too many variables. We do allow patients who are in some of our clinical trials to take Rilotec, which is FDA approved, or Radicava, which is FDA approved, in addition to whatever else they might be taking. So that's sort of my take on it.
Yeah, I mean, I share your frustration. I mean, ALS, in my opinion, doesn't get the attention that the cancers out there do. I mean, there are immunotherapies for cancer. My mom passed away of cancer 2 years ago. She was a 3-time breast cancer survivor. The last time it came, it was so serious. There's no immunotherapy out there that probably, could have really helped her. I see these commercials every single day about this immunotherapy for cancer and so forth. And I think back, could that help my mom 2 years ago? I wish she was alive to find out. But, it's important. I mean, if I go around being angry and saying, you know what, and being bitter because my mom passed away of cancer, I think that would really, really affect me a lot. But I've got to remember her and cherish her memories. But I completely agree. I mean, there are immunotherapies out there for cancer. ALS, is maybe the redheaded stepchild. I don't know. But there's more attention that needs to be made for ALS, whether it's on the, on the congressional level, where, there's a major push for, kind of like Joe Biden's, a moonshot to cure cancer. There should be a moonshot potentially to cure ALS as well, or at least figure out how we can stop it or slow the disease progression. So.
Time for another question, or?
Is our microphone dead?